The purpose of this tutorial is to demonstrate how to use the various features of the heat map to visualize differential gene expression from GEO datasets on Xenbase. The heat map visualization tools are accessed via the GSE pages. To get there from the Xenbase homepage, click on the Expression tab at the top and follow the GeoData at Xenbase link. Or go to the Gene Expression tile in the middle of the Xenbase homepage and follow the GeoData at Xenbase link. This will bring you to the GSE search page. Search for your GSE of interest and click on the GSE link to go to the GSE page. Scroll down the page to view the sample table. You will see that each sample is listed in a row and indented under its respective control. To generate a heat map visualization, you first select the samples you wish to view. To do this, check the boxes next to each sample and click the Compare button at the top or bottom of the sample table. This will bring you to a log2 fold change heat map visualization. The colors for the heat map can be changed by selecting any of the options in the drop-down menu. Below the chart type and color options are the default filters applied to generate the gene list visualized in the heat map. These values can be changed simply by typing in a new value and clicking Search. Above the color scale, you can see how many genes are returned based on the filters. In this example, the default shows 30 genes. However, there are 122 genes that meet the filter cutoff. To view more genes, change the number in the Show Top Genes box and hit Enter or click Search. Now we can see the top 75 genes in the heat map. Note that as you move your cursor over the cells of the heat map, the gene and sample are highlighted and associated data are displayed. If you mouse over the gene symbol at the left, you have the option of going to the Xenbase gene page or viewing the model in JBrowse. Genes in the log fold change heat map are ranked by log2 fold change based on the sample from which the gene list was generated. This sample is shown immediately adjacent to the control column. It can be changed in the drop-down menu next to DEG list from. Here you can see all of the samples selected from the previous page. Choose which sample you would like to use to generate your new gene list. The log2 fold change of the same genes in the other sample will be shown, but these are not ranked. After you have generated your desired gene list and heat map visualization, the Save to CSV button allows you to download the gene list and the underlying data that generated the table. Similarly, you can save the image to an SVG. This is an example where there is one control. You can also view samples that have different controls. To do this, return to the GSE page by clicking the link at the top. For this particular GSE, there are two controls. There is an option to select all samples at the bottom of the sample table. You can do this by checking the box and clicking Compare. Now you can see there are two controls in the far left columns. Each control has a letter When you hover over the letter, you see the associated samples highlighted in red in the other columns. Note that there are a number of chart types that are available to visualize differentially expressed genes. To change the chart type, click the drop-down menu. Other options include TPM heat map, in which top genes are ranked by log2 fold change. However, the color scale represents scaled TPM. The FDR heat map ranks the top genes by FDR. The color scale represents the FDR values. The gene symbol heat map simply takes the data from the gene list you are currently viewing and organizes it alphabetically by gene symbol. In this example, it takes the FDR ranked list and organizes it alphabetically. Two other types of visualizations are parallel coordinates and a hive plot. There's also an option to analyze a custom gene list. To do this, click on the custom link next to all. Input a list of genes separated by commas, spaces, or line returns. Once you have input your list of genes, click the search button to generate a heat map. Genes not found will be displayed in red at the top. The default FDR is 1. This will display colors for all genes in the heat map. Decreasing the FDR will fade the colors of the genes that do not satisfy the FDR cutoff. Note that an FDR less than or equal to 0.05 is considered statistically significant. Columns are organized by changing the sample in the first column experiment drop-down menu. This determines the order of genes based on their log2 fold change in the selected sample. The color scale displays scaled TPM or log2 fold change. This can be changed in the chart type drop-down menu. This completes the heat map visualization tutorial.